So today we're joined by Dr. EuanMcDonnell from the Computational Biology facility, and he's going to be talking to us about bioinformatics and the curse of dimensionality.
Yeah, thanks. Yeah, so I'm Euan, I'm a bioinformatician. I'm going to talk about the kind of field I work in and how this issue of the curse of dimensionality, high dimensional data, and kind of the issues it poses for the sort of analysis I tend to do, and then also the kind of how we try and resolve this with a bit of an example of kind of case study from my own research to go into this. So just a bit of background of who I am. So I'm a bioinformatics data scientist, as Alex said, based out of the Computational Biology facility, or CBF, based in the University of Leeds. Work on a range of kind of long, medium, short-term projects. Range of collaborators includes academic collaborators and also some clinical collaborators from hospitals around Liverpool and various other institutions. Host a range of R courses. So as a CBF, we run some R courses and also do some kind of more extraneous kind of courses with different research councils. So there's a bit of background. I started off with doing an integrated BSc and Masters in microbiology. This is at the University of Leeds. During that period, did a few wet lab placements, really during the summer. Realized I wasn't very good at it, kind of hated it. So I shifted to doing more kind of bioinformatics, computational, kind of dry lab approaches. And that was during my Masters, I did a kind of hybrid project where I was looking at cleavage sites in biological molecules called RNA molecules, if you come across them. In what's known as transcriptomics data, which I'll delve a bit more into later. But it was during this period I learned the kind of like computational biology, or broadly kind of computational analysis, kind of bread and butter, things like Linux, things like the command line, Bash, and also a bit of Python. I then did a PhD based at Leeds as well, but that's specifically in bioinformatics, and that was looking at regulatory networks in the context of an oncogenic herpes virus, so cancer caused by the herpes virus. And it was during this period that I really kind of started learning the kind of real fundamentals of computational biology. So these, statistics, computational biology, R in particular, the R programming language is pretty much universal within bioinformatics, and a few other things like pipeline managers like SnakeMake. So just quick examples of some of the projects I've been working on at the moment or I'm going to finish. So I'll do things like one of them in the middle is looking at changes in gene expression, so kind of gene expression profile responses in neonatal patients with and without treatment with arginine. The idea is it would these premature neonatal patients, the idea is that it would improve outcomes for them. And we're trying to see kind of the effect that it had in terms of the actual biological effect rather than clinical effect. Recently published a paper on trying to relate. So we know that there are a lot of genetic variants that contribute to the risk of developing things like osteoarthritis and other kind of conditions like that. But what's not quite known is exactly how this risk can manifest, what actual mechanisms means that if you've got these genetic variants results in you kind of increasing your risk. And what we showed is some of that affects basically certain DNA modifications which affect the expression of genes, but particularly during the development. So it's during fetal development that this stuff kind of manifests, during that period. Some other projects I've worked on as well, kind of more small scale, and these with hospitals. So one of them was working with Alder Hay and looking at changes in craniofacial measurements pre and post surgery to kind of assess the effect of them, the effectiveness of the surgery, kind of over time, before and after, that kind of thing. And then the final one is looking at key microbial indicator species and their relationship to intestinal inflammation and other clinical covariates in neonatal patients. So just background on the CBF is what we do. So we kind of work with a range of scientific partners, collaborators around the university and externally. We do some training workshops, things like programming bioinformatics, mostly our programming courses, but we also do more specific training courses as well. Bulk of what we do is data analysis. That's kind of bulk of what I do, particularly work on things like omics and multi-omics. And that's the sort of data I'll talk about, but we also develop pipelines that do kind of more standard statistical and machine learning analysis. We develop some software and databases for bespoke problems, as well as offering some high performance computing suite software as well, solutions. And it's not so much something I do myself, but we also do protein bioinformatics. So this is kind of structural biology, trying to predict protein structures and things like that. And then also infer protein function from that. And then this is the team. So it's Andy and Eva, the directors, John's the manager. We have some associate directors who are kind of one foot in the CBF, one foot out. Dan, Jamie, and Francesco. We also have Tony, so kind of main senior software engineer. And then we have the range of data scientists as well, which includes me, but a range of other people. And we're currently also kind of taking on some more to the team as well. So we're kind of growing a bit. And we're also part of, so if you've not come across this, the idea is kind of Liverpool shared research facility. So with a lot of, particularly bioinformatics, it seems to be sort of computational biology, but I don't know if it's similar for other kind of disciplines as well, as you get different kind of almost like nuclei of expertise kind of forming in different departments, but a lot of times they don't kind of interact. So you lose the ability to kind of complement each other. So the idea of this initiative, which started off by the university, was to try and kind of have a bit more of a framework here. So the idea is maybe, so we're in the strand with multi-omics, that's the sort of data that we work with. And we have kind of sister institutions like the Center for Genomic Research, Proteomic Research, Metabolomic Research. So these guys generate and do some analysis on those particular types of data, so genomics, proteomics, metabolomics. But we're able to kind of, rather than us generating in-house, we get them to generate it, then we're able to apply our expertise, which is the more kind of integrative analysis and stuff like that. So just after the introduction, so talk layout will be introduction to bioinformatics and omics techniques, as well as the kind of associated data types, the kind of broad field of bioinformatics, tiny bit of history, the sort of technologies that we tend to work with, and kind of the data types that result from how those technologies generate the data. Some issues of the high dimensionality of such data and how this relates to the sort of modeling we have to do. And then a case study of my research that kind of walks through some of these how some of these challenges and then how we kind of try and deal with them. Yeah, so just start off with this. So it's a fairly broad term, it's often synonymous with computational biology, and I say bioinformatics can end up being a bit of jack of all trades, where you're doing lots of different stuff, but people definitely do specialize in these days as well. But the foundations arose as early as about the 1960s with kind of computational approaches, in particular to do things like analyze protein sequences and then later DNA sequences. But it's really the kind of 21st century that saw the kind of convergence of a lot of disciplines towards sort of a notion of information, and particularly information in biology, but it required a lot of understanding, a lot of kind of insights from other fields like biology, chemistry, physics, maths, and computational science. So I think what I would emphasize in a definition for bioinformatics, although people could probably disagree with me on exactly what they're doing, the exact sort of bioinformatics that they're doing, the sort of stuff I do is it's kind of an emphasis on the management so it's the informed bit of the bioinformatics, that kind of informational bit. And it's at some point, just the emphasis on management, handling, processing, QC, and analysis of such large like big data sets, right? That's one kind of big aspect of it. It's a kind of practical aspect of that. But it's also the kind of a more, I guess, sort of a sort of perspective on the type of analysis and the way of understanding biology is more of a kind of systems of information, how these things kind of spread between the different systems within biology, and the kind of, it merged out of something called systems biology, which is kind of a way of understanding biological systems in totality, in a kind of top-down sense, rather than a kind of reductionist querying every single protein or something like individually. A lot of it relates to, so you'll have certain molecular observations that you're taking, quite low scale stuff. So classically, this could be genetics, genotype, and how that relates to phenotype. So, your genetics influences your hair color, kind of understanding that mechanism. But more broadly, you might be investigating certain cellular metrics, certain cellular processes that are going on, and how that might relate to, a phenotype in the sense of like disease, right? Whether this regulatory system becomes dysregulated or chaotic or has some issue with it, and how that results in pathology. So this is kind of one of the insights that kind of came out, I think it was about the 1950s, 1960s onward, is this idea of kind of information flow in biology. So I think this is sort of guys like Erwin Schrödinger kind of came up with, or initiated some of this stuff. And there were some meetings relevant to this, but it's kind of developed over the years. But the idea was that, like, analogously to something like natural language, right, which is kind of comprised of individual characters, these kind of get assembled into words, and then these then are assembled via some overall structural rules into a sentence. It's once you get those sentences, once you get those kind of higher order language, you have meaning, you have some kind of function, the thing is able to express itself in some form, or the human maybe is able to express itself. So and analogously to that, sort of biological sequences, in this example, proteins, are formed of individual amino acids that come together in sequences, and then through some kind of rule set are structured into a protein that is, a physical thing that has shape and has structure. And it's only once you have that kind of structural formation that you get the kind of function, the expression of that protein. And then just as a side note, so this particular kind of question, this thing of how do you go from, polypeptide sequences, amino acid sequences, to, a functional protein and understanding the structure of a protein is the kind of the topic of probably one of the most important algorithms in molecular biology and bioinformatics the past 40, 50 years, really, which is AlphaFold, which you might have heard of. It's a deep learning model, basically, for folding proteins, of learning this relationship between sequence and structure, and thus function as well. And it's been very useful. I don't know too much about it, it's a bit out of my field, but I know people who are working with this kind of stuff. So another idea of kind of information flow is the idea of the kind of central dogma. So it's maybe throwing back to kind of AQA biology if you did it. But the idea is that kind of the core way that the DNA, that the human genome, that any genome, sorry, is able to kind of transmit its effect is this idea of kind of information flow through different states. So DNA made up of a sequence, it has some kind of that sequence, specific, it encodes things like genes or stuff like that, that encodes the genetic information that needs to be kind of expressed to have kind of an effect in the cell. Traditionally, the idea is that this DNA undergoes a process called transcription, forms an RNA molecule, then is translated into a protein, which forms the protein, and then this kind of has the influence in the cell. And this kind of process is kind of linear, and it's like kind of the process of gene expression. But this is kind of analogous to kind of information flow. So the way that I like to think about it is basically DNA encodes a certain amount of information. It's transcribed into RNA, which is then able to kind of leave the nucleus where the DNA is stored out into the cytoplasm, where it can actually have a bit more of an effect. But the kind of code itself is the same. The actual information is the same. It's just in a different format, right? It's still the nucleotide code. It's sort of just in a different kind of format. It's able to leave the nucleus. But when you actually have the process of translation, that's actually converting it into a different code. But it still encodes the same information, right? You're kind of translating from the nucleotide code to the amino acid code. This is a bit of an oversimplification. So the past like 50, 60 years have shown that there's very high levels, complex regulation going on here. But broadly, although these all add complexity to the system, it's still representing different forms of information flow and how this kind of moves up and down this kind of system and how it kind of has the ultimate effect of the cell kind of undergoing its own kind of function. And another thing to emphasize as well is the kind of complexities of cells. So, the classical view of a cell of a kind of biological system is kind of just a bag of goo with some proteins and DNA in it. But we actually know that it's actually very, very complicated. They're much more complicated than that. There's huge ordered structure in there. So it's not only structure, you have kind of dynamism going on there. So for instance, the cytoskeleton forms a lot of the structural kind of features of the cell, but it is also quite a dynamic thing. So it's involved in transporting things about the cell, but it also involved in movement, and things are constantly changing over time. On top of that, you have different organelles and stuff like that. So, things like mitochondria, chloroplasts, that kind of stuff. But these kind of allow for complex chemistries going on in the cell and quite a kind of dynamic mechanism. But also just the sheer kind of number of things going on there and the sheer kind of high order structure that's in there means that a lot of methods that were kind of more geared towards investigating things in a kind of individual component, in a kind of component-wise sense, were relatively, miss this kind of information, this kind of overall structure that's very important to how the cell actually functions, how these things actually operate. So on a kind of more practical level, the real thing that kind of got bioinformatics kicking off is, again, this idea of kind of different kind of technologies coming together. The main development was what's known as kind of high throughput technologies. The importance of these is basically, so rather than interrogating individual components in a kind of specific manner, i.e. a biased manner, we're going to try and just look at all the entities, all the biological things. So it could be genetics, the genome, sorry, it could be the transcript, and it could be the proteome, which would be all the proteins basically in it, or it could be a bit more kind of abstract than that. So it could be biological processes like metabolic reactions or something like that. Just the idea is looking at everything in a high throughput manner, kind of everything all at once, and then kind of in a way that's not biasing to any one kind of subset of it. You're trying to get an overall global picture of it. Another important thing was prior kind of work beforehand. So a lot of this analysis was kind of only able to be done with a lot of painstaking working on just doing things in more low throughput manner. So a classic example of this is the Human Genome Project. I think it took 10, 15 years and it cost over £1 billion to do, sequencing tiny bits of the human genome in parallel. Human genome is 3.2 billion nucleotides, so it took a long time to do. But once we had that data available, it meant we could do a lot with it. So it was a very kind of a seminal moment to allow kind of later things to emerge. So the sort of bioinformatics that I do, and a lot of bioinformatics in general, tends to be focused on what's called omics data. So this kind of refers to like the something ohm. So this will represent, as I was kind of saying, the entirety of that one thing. So for instance, genomics might be one you kind of come across, which is looking at the entire, genome of an organism, or multiple genomes sometimes. If you're looking at something like microbiomics, which is looking into multiple bacteria within a kind of population. You might have things like metabolomics, looking at all the metabolites. You might have proteomics, again, looking at all the proteins, or transcriptomics, which is looking at all the RNA, so the expression of those genes, that kind of intermediate between DNA and proteins. And most of these these methods that were developed during this time are known as kind of shotgun approaches. And I'll explain why that is in a little sec, but these kind of relate to things like microarrays and next generation sequencing. There are more modern kind of ways that don't rely on this kind of shotgun approach, where you kind of what you're doing is basically fragmenting your stuff before you're trying to sequence it, or before you're trying to quantify, like physically breaking it up, basically. There are whole molecule ways of sequencing, things like nanopore you may have heard of, but it's relatively new and it's outside the scope of the sort of stuff I do, at least for the kind of analysis and the questions that I have. So this is my kind of broad, I'd say, like generalization of what shotgun omics techniques are like. So you'll have some kind of a sample, you'll have some kind of unseen, entities, unseen proteins, let's say, of unknown abundances and unknown identity. You're looking for proteins, so there are ways of specifically extracting proteins without being specific about it. But then what you do is you take these kind of purified protein set or purified biomolecules, and you'll fragment them. This is where the kind of the shotgun idea comes from, where you're kind of fragmenting them. That's because it's quite difficult to deal with quite large molecules. Proteins could be thousands of amino acids. Transcripts can be hundreds of thousands of nucleotides. They can be quite difficult to work with for various reasons. But then the other side that kind of was important for this, as well as that kind of technology, was these kind of information of these kind of sequences, these kind of prior information that was developed. So I kind of mentioned the Human Genome Project. So this is literally just a string of characters, 3 billion nucleotides-ish long, that tells you, the reference, the sort of consensus genome of the human genome. And by kind of aligning the two, so if you take your sequences of your kind of fragmented thing, if you do it in the right way, you'll be able to, in theory, uniquely align them against the human genome or against that genome you're comparing them to, which again, it's just aligning two sequences, basically. So by looking at where it's aligning on that genome, on that kind of that cat string, with known kind of annotations, so you might know this part of the genome is a gene, that kind of idea, you're able to not only annotate, or sort of find the identity of that fragment, but you also have to quantify in that way. So you're kind of getting both the identity of the thing you're looking at and also the quantification of it, and that kind of generates your features effectively. And yeah, so I was talking about proteins and DNA, but these can really be anything arbitrary. There's thousands of different types of omics technologies now, so this is quite a general topic. But I will focus on just two of the main technologies that certainly I've worked with, but generally are the kind of main technology, at least for working with DNA or RNA or kind of gene expression, that kind of stuff. So the first would be microarrays. So these are literally just kind of glass chips, where on each of these chips you have little kind of dots. And these dots, if you kind of look into them, are kind of comprised of having little kind of DNA probes. What these have is they have a fluorophore on them, so something that's able to luminesce under the right wavelength of light, but also they have a sequence that is specific, so binds specifically to a region of the human genome, because you have this kind of complementarity that DNA has, where the sequence, a certain sequence will bind to another sequence. What you effectively do is you take your fragments, you take your library, and you wash it over those sites, over those probes, and the fluorophores are set up in a way where upon you only get fluorescence, ideally anyway, upon binding of these fragments. And then you're able to measure the intensity of that fluorescence, and that gives you an idea, whether some kind of wavelength of light or something like that. And that gives you your measure of abundance, as well as the identity, because you have a kind of index of where each of these spots are and what they correspond to. But I think the way to think about that is, so this is more, when I talk about this kind of alignment that you're doing here, this kind of comparison between the reference and the actual sample you have, it's more of a physical alignment. You're kind of exploiting the biology there that you know about in order to do this kind of quantification. But then with next generation sequencing, or NGS quantification, this is different, because this is a later technology developed about 2008 onwards. This is where you have your kind of fragments, and you do this process called sequencing by synthesis, where you're able to kind of sequentially wash the fragments with different nucleotides, with different fluorophores. Then based on what fluorophore is, what wavelength of light is being emitted, you know what the base is that's being added. In this way, you generate a set of reads in silico, which again are just literally just strings of characters, A, G, C, T, the nucleotide code. And so rather than doing this kind of physical binding, You then use various algorithms to map, or what's often known as genomic alignment of these sequences to, again, the human genome. But by looking at whether things bind to and overlap with, looking at the sort of genes and things they end up under, you're able to get both the identity of those reads, but also the quantification of those features as well. So that's how you kind of get your data out at the end. And the features end up being those kind of units, whatever you focus on. So whether they're genes, transcripts, anything like that. But as with anything, the way that these processes, the way the data is generated from these processes result in the kind of define the resultant data distributions. So for microarrays, because it's kind of a luminescent signal, it's continuous and generally it can be assumed to be Gaussian. But for NGS data, for this kind of this read, this in silico data, you're literally quantifying individual reads mapping to different regions. And that means it's count-based or it's integers. And generally this is assumed to be modeled by things like negative binomial or Poisson distributions. And yeah, just a bit about the actual data itself. So the sort of data we deal with tends to be tabular. And the convention is rows are your features, columns are your samples, and these could be in different groups or replicates or cells or something like that. And for microarray data, it's continuous. Usually tends to be Gaussian, and then for NGS data, it's integer count data. But this is just convention. Depends on the analysis you do. You can always transform them. So now I'll talk a bit about how the high dimensionality of this data can be an issue and how it kind of relates to the sort of modeling we choose to do. So as I was saying, so the idea is to measure the entire ohm, proteome, whatever, of the thing at once. But when you actually start looking at some of the figures involved for this can be quite problematic. So as an example, humans contain over 20,000 protein coding genes, up to 60,000 if you include things that don't strictly encode for proteins. So these are things like non-coding RNAs, a range of other stuff as well. Of this kind of output of the genes, there are up to 250,000 transcripts because you have various processes like splicing. That means you get lots of more different transcripts within the cell as well. And of these ones that can be converted to proteins, because you have different modifications and various stuff, you can end up with hundreds of thousands of proteins. So the human genome is 3.2 billion nucleotides, roughly thereabouts. And of the people that we've sequenced or genotyped, so we've looked at kind of key genetic regions on them and looking what varies between these people. I think it's about 30 to 50 million people have either been sequenced or genotyped. So at least 100 million locations have been on the human genome. So these are individual nucleotides have been shown to vary across that population. But that's only basically constrained by how many people we've done that on. If you did the whole human genome, it's probably way more. It could be really the whole thing. And obviously that itself is constrained by how many people you have as well. So the broader your population, the more diverse is it, the more genetic variability you'll have. So needless to say, a lot of this can get quite high dimensional, especially when you're dealing with genetic data with these kind of nucleotide variation data. And this kind of goes, also there are some factors to consider here though, is that you may not identify all features at once. Some is just due to technical reasons. So inherent sampling variation, tends to be assumed to be a stochastic process where you will miss certain things due to fluctuations in the system you're measuring. Issues of limited detection. So the instrument you're using might not be sensitive enough to pick up individual very poorly abundant features. But also that relates to the dynamic range. So if I'm trying to measure proteins, the far end of the scale is one arbitrary unit, and the other is a billion arbitrary units, or 10 billion, let's say. With the technologies that we have, it's quite difficult to accurately quantify each end of those scales. So you tend to have to kind of window a little bit. You have to have some, you have to kind of cut off the tails of the distribution in some way when you're doing this kind of analysis. That's particularly true for proteomics or anything you're using like a mass spectrometer for to quantify stuff. But there's also biological reasons as well. So almost by definition, different cells, different tissues, and different contexts will have different biological features present. So for instance, the gene expression profile, so the entire kind of sum of the things that are expressed in any one cell at any one time kind of defines the output of its behavior. And differences in that is almost what you're interested in looking at. But by that very nature, you won't pick up everything at every single point or in every single condition because they won't be being generated or they won't be there because they're not relevant to that condition or something like that, or relevant to that context. But it's still frequent to sample hundreds to millions of features, still a huge issue. The other side that makes this quite difficult as well is the fact that data collection and generation can be very problematic. So the first is it's very time consuming and expensive. Some of these technologies take a lot of time in terms of researcher hours. I've heard horror stories of people doing sort of 96-hour time course experiments where they're having to kind of go take samples at like kind of regular intervals across that whole period. But it's also the kind of the quality control, the processing, buying the kits from companies can be very expensive relative to other stuff we do. And yeah, so certainly it's quite expensive as well, where like the kind of reagents you need are quite expensive. It can take a while to collate everything together. Another issue is ethics. So obviously we're dealing with biological organisms, potentially human samples as well. There are some, depending exactly on what you're collecting can limit how many samples you should really take. For instance, you might go around and collect loads of leaves from trees. That's probably fine to take quite a lot, but taking cerebrospinal lumbar punctures from kind of neonatal patients, you really don't want to take more than you really need to for the context that you're working on. So a lot of kind of biological projects, you should really be doing things like power analyses, basically saying, I'm only taking as many samples as I would need to see the effect that I think, or as many as I think to see the effect that I'm looking into. And then obviously that relates to things like animal models as well. You want to limit usage of animal models for ethical considerations as well. Other issues are just just issues with like the rarity of the condition. So certain conditions are just by their nature very rare. They could be kind of very isolated kind of genetic variants or stuff like that. You're practically constrained basically by the population that has that kind of variant or has that sort of condition or that disease. So that constrains, the number of possible data points you could be looking at there. There's also just the effect of can be quite difficult to collect some samples, going out into the compost heap and looking at the microbiota of earthworms is probably a lot easier than looking at the microbiota of like Bengal tigers or something like that. It's challenging to do that in a way that it might not be for other contexts. So generally, when we're talking about this sort of analysis, we've often got hundreds to thousands to really tens of thousands, hundreds of thousands more features than we do samples. So broadly, this isn't a problem unique to bioinformatics. It's general to a lot of machine learning is you tend to not have as much data as would be beneficial under your standard kind of statistical frameworks. But it is kind of noticeable for its kind of ubiquity and prevalence in bioinformatics. Broadly, this corresponds kind of to what's known as the high P, small n problem. P meaning you've got a lot of features, N meaning you've not got many samples. As far as I know, other examples where this is an issue are things like image analysis. I guess where you're limited by the number of number of images you're able to take for certain things. And I assume it's quite high dimension and the sort of number of pixels or wavelengths of light you're looking at for the images. Document classification, astronomy, and things like atmospheric science as well, where you've got quite complex systems that you're looking at with lots of features, but you can't take that many data points or that many measurements over whatever period of time you're looking at. And yeah, this kind of issue of, this relates to the issue of sampling a kind of high dimensional space. And this is referred to kind of the curse of dimensionality. And this is where you kind of, the sort of possible combinations of features that you could sample gets exponentially bigger as the number of features go up, the feature space increases. And it's in a way where if you're not collecting enough samples and you're not doing it in a sensible enough way, you're effectively going to only focus in on one subdomain of that possible feature space, which means, any kind of models or inferences you make off of those samples may well not generalize to the rest of the feature space. You're effectively kind of overfitting to that kind of subdomain in many ways. So just some common solutions we tend to use to deal with this. So one is make some assumptions about the data you're dealing with, the data type and its distribution. So this would be kind of enforcing some kind of parametric model. So this is, I'm going to assume based on some prior theory, that this model could be modeled effectively with Gaussian data, or as I was saying, with like negative binomial or Poisson distributions, these kind of making these distributional assumptions. This can be very good because it increases your power and increases the kind of toolkit of analysis that you can do. But this comes at the cost of introducing bias to your analysis. So you're sort of imparting an understanding, effectively a simplification. You're applying a model in some way that may well not account for the entire variability or the entire kind of complexity of the thing you're looking at. Other very common ways as well, doing things like cross-validation, so just kind of splitting your data up, building models in individual sets of the data or dropping samples or something like that. Try and kind of artificially expand your sample set or kind of sort of in silico model what it would be like to take lots of different samples across. You kind of trying to increase your sample set almost kind of artificially in some ways. But more broadly, the kind of goal to tackle this sort of analysis is to identify or produce a set of features. So if your initial features P, your input, that are smaller than a set of features that's smaller than that initial input P, but that explain most the association or the difference or the separation between the outcome of interest. Basically, you're filtering down your features to focus on those that are the most explanatory or contain the most information in the kind of task you're trying to do. So very simple one would just be do univariate feature selection. So you have a list of features, a set of measurements, you have rows and columns. Per feature, per row, just do some kind of test statistic, calculate a test statistic of some kind. Could be do linear modeling, could be more non-parametric than that. But the idea is you then kind of basically calculate something that you're then able to rank these features by, apply a threshold and take those forwards. That's obviously quite simplistic and can miss a lot. Another common thing would be something like dimensionality reduction. So this is when you take a set of features and you're basically transforming them into a lower dimensional space with the idea of you lose interpretability in these features, but you retain as much information as possible about the initial features you had previously. And then the other thing that tends to get used a lot as well is regularisation. So this is this kind of classic kind of method that's done for things like lasso regression or ridge regression, but often is also can be applied to pretty much anything you're trying to estimate some kind of objective function with, where you apply some kind of a penalty term to the thing you're trying to the kind of likelihood or something like that, and that aims to kind of shrink the likelihood of things that are potentially less interesting, and then promote the likelihood or shrink less the likelihood of stuff that you're more interested in. In a way, it's kind of like implicit univariate feature selection, except for the fact that when you're doing this, you're considering all features. So it's a multivariate kind of approach. But there's also kind of more or less standard, more kind of biology-y ways of doing this. So one thing you might do is, so based on the underlying understanding of biology, so you might know that certain processes are involved in this kind of in this condition or this disease you're interested in. You might know things like glucose metabolism is very important to this kind of situation. So you may pre-filter the features initially by what are known to be associated with those processes. And there are more formal databases, so it tends to be kind of gene ontology or pathway databases of sort of genes or biological entities, can be proteins, can be metabolites, anything that are involved in some kind of biological process. So these can be, reactions, like I say, glucose metabolism, but it can also be, certain diseases, so certain viral infections, or it could be stuff like the cytoskeleton, so physical structures within the cells and stuff like that. And then there are also more kind of complex ways of integrating prior information under kind of Bayesian frameworks. So kind of saying I have some prior understanding of how I expect my data to be distributed, or I expect to kind of the idea is basically of my possible feature space. Can I put some kind of constraint on this where I can kind of only solve within those kind of constraints? Ideally, meaning I arrive at a more robust and reliable conclusion, but that obviously depends on how good your prior inferences are. So finally, I'll just talk about kind of case study from my own research. So this is this will walk through different example, at least one example of each of these kind of methods and kind of how I've kind of dealt with it. So this project is one that's still somewhat ongoing, but this kind of first phase of the project has been finished. So this is looking in to identify or learn protein biomarkers, so kind of proteins that discriminate between different pediatric inflammatory conditions. So the idea is these certain conditions are quite clinically similar, or certainly difficult to distinguish based on clinical means alone, various symptoms or various classical ways of measuring certain things. It's quite difficult to do, and it's quite costly for doctors to do. It takes a lot of time, and it's also has quite a high rate of false positives as well. So the main idea is to try and distinguish between this one condition called chronic non-bacterial osteomyelitis, or CNO for short, and 3 other similar inflammatory conditions. So infectious osteomyelitis, which is effectively equivalent, but caused by like specific bacterial infection of the bone, effectively. Crohn's disease, and then also oncology patients, and then also kind of contrast these with healthy patients as well. So we want to distinguish this condition from healthy, and then also distinguish it from other mimic conditions that it could be confused with. Yeah. So this is the kind of broad pipeline that I've been using with this. So this is looking in serum samples, so it's blood samples taken from patients. We take about 170 from pediatric patients, from various patients, both at Al so the Children's Hospital here in Liverpool, but also a few others as well, like Dresden and Vanderbilt University. These were sent for untargeted mass spec-based proteomics, similar in idea to the shotgun approaches I was talking about, but instead of dealing with DNA sequences, you're fragmenting proteins. You're then applying some kind of charge to them, and then quantifying the kind of peptide spectra you get at the end. And the kind of way that you're annotating them, I guess, is, you'll have some kind of unknown fingerprint, is you've got prior kind of databases of peptide spectra. So you're kind of matching it in that way, but it's a bit more of a kind of pattern spectral mapping than, sequence and character kind of mapping. But broadly, the idea was you kind of get the raw data out of this. The resultant data can be assumed to be Gaussian, can assumed to be normally distributed. But you take your kind of raw data, process and quantify it. So that's getting your features and getting your abundances. You then do various quality controls, so normalization to try and control for sample-to-sample variability, various quality control metrics. You tend to have to do imputation with this method, so I'll be quite short on this because it's a whole kind of other topic, but is the idea of missing values. So this ideally, in the ideal case, tends to relate to this issue of limit of detection, where certain proteins will be so poorly abundant that your mass spec machine won't be able to detect it. But their abundance is going to vary by samples. So in some samples you might see it, in other samples you might not. You can just discard them, any with missing values. And you have to consider them as missing, not zeros. Generally NA if you've used R at all. Or otherwise, you can kind of you're implicitly biasing the distribution, right? You're effectively zero inflating it if you fill in these missing values with zeros. But the kind of choices you make with doing this imputation can have consequences. I'll show you a little bit later on. It tended to not really have too much an effect, but it is something that's worth considering in this sort of analysis. We then proceeded, once we're kind of happy with some of the quality control steps, although this process is obviously quite kind of cyclical. You're kind of doing UGA, you're changing your QC steps, you're kind of constantly doing this until you arrive at where you think is the best. Did some just standard kind of analytical methods, so things like principal components analysis or PCA to kind of quantify the data, see how the samples were laid out. Then performed a form of kind of univariate feature selection, so taking about 487 proteins forward. Not the most high dimensional relative to the samples we have, but it's still problematic for the kind of tasks that we want to do. But we wanted to kind of pre-filter these proteins because we realized we could kind of do this in a way that kind of potentially putting in that many proteins, 500 proteins into the lasso algorithm wouldn't work out so well, would result in relatively inconsistent results. So we wanted to do some form of kind of feature selection initially. But then once we've kind of filtered that down to about 187 proteins, we plug this into a kind of multivariate regularization kind of framework to select for proteins, and then do some classic, kind of machine learning model testing, test training on orthogonal validation data sets. But the reason I kind of wanted to talk about this project was I think at each of these stages, I do at least one of these different filtration methods. So I thought it would be quite a good example of this kind of stuff and how it integrates into a kind of bioinformatics pipeline. So yeah, the step I kind of did, or one of the steps I did principal component analysis was here. So this is a PCA applied to all 170 proteomic samples across all 487 proteins. So I've not done any filtering here. And I'm showing a bioplot of PC1, PC2. I'm showing the individual samples. I'm coloring them by the conditions. The main thing to see here is that it's quite hard to separate the different conditions based on these proteins alone. So you'll see the orange ones there are quite interdispersed with the different subsets of the data. So it's the healthy ones in green, some of the pink ones, the Crohn's, and also the oncology patients quite interdispersed. This indicates that there's a lot of confounding or at least redundant information going on there that's kind of masking the signal that we're interested in distinguishing between these samples. So this is, a classic multi-group classification problem that we're trying to do here. And you can see why when it comes to actually trying to, for our overall goal, which is to be able to provide proteins that can go forward to develop a test, just looking at the entire proteome based on this understanding is not really providing us a lot of information, a lot of accuracy in being able to distinguish between these classes. The next stage was to do univariate feature selection. So because we're particularly interested in comparing C and O to the other conditions, and we don't necessarily we don't care if it's different between all conditions at this stage. We're just kind of trying to pre-filter things to make the actual selection run more smoothly. It would be kind of inefficient to do individual pairwise contrasts, individual tests between each of the conditions. So what we settled on doing was setting up 4 contrasts. So CNO to the Crohn's, CNO to the healthy, CNO to the infectious osteomyelitis, CNO to the oncology, and then fit an ANOVA model to test if any of those contrasts significantly vary from 0. So that basically tells us, filters our proteins where they vary with at least one comparison between these conditions. And yeah, this is filtered down to about 187 proteins to take forward for multivariate selection. This is still kind of too many to practically test via kind of low throughput, high accuracy methods. And so it's kind of quite difficult to develop this into a kind of reasonable diagnostic test. Generally, these kind of data types, although they're quite powerful in terms of the breadth of the features you're able to look at, any one feature, you don't have that much confidence in them. So it's not really practical to use those kind of methods for diagnosis, at least for the sort of funding we had to do for something like this, but also too many proteins to do via more low throughput methods that are more accurate in detecting things. It's quite difficult to do that with lots of proteins. There's lots of interactive effects and things going on there. But also by kind of just solely focusing on univariate filtration, we potentially miss important kind of multivariate relationships between the proteins as well. Because a lot of processes that happen in biology can happen due to the kind of concerted effort of little changes, but consistent changes. So say if I'm thinking of a certain process being upregulated, maybe say like glucose metabolism is going up. It might not be that every single protein is going up loads individually, or just one protein is going up. It's that all of the, say, 20 involved in it are going up by a little bit. And you can argue whether that's important or not, but a lot of people, that kind of I guess enrichment of changes is often considered to be very important in this kind of field. So this is moving on to the kind of multivariate selection process that we did. So I should say also prior to doing this, we also did an 80%, 20% train test validation split. So we limited data leakage between the two when it came on to testing the validation data. So taking forward that 80%, did the univariate filtration, did 10 test train splits on the data. So 80% train, 10 test, plug this into a lasso regression framework. And rather than just running one round of lasso regression, we did 100. The idea is to kind of this issue of if you start off with if you just do one round of kind of lasso regression or any kind of problem solving this way, there's variability in kind of where you're starting, where the model's starting and where it's ending. And so we can end up with non-unique results. So we're trying to just kind of account for that variability in the kind of modeling that we're trying to do. But we defined that, so that's operating on selecting individual proteins from that kind of set of 187. But then we then filter those proteins, so across the 10 splits based on whether they were present in greater than 75 of these repeats of these runs of the lasso regression in over 7 splits of the data. So that's kind of what this plot on the left is trying to show, where the individual box plots, the kind of columns along the x-axis represent the individual proteins. The ones that are in blue are those that pass those thresholds. So basically there are 7 dots. So each of those dots, each of the box plot represents the distribution of the runs across for each protein across the different splits of the data. And the idea is that you have more than 7 splits above that line. And they're the ones in blue. The ones in blue are the proteins that were selected in this case. And so we then took forward this set of biomarkers depending on range from about 15 to 40 proteins. And then use these as features in a random forest model to then predict the classes. So we then kind of take it on the test data, so that we kind of held out, and then we test and calculate various model QC metrics. And yeah, then obviously we kind of we didn't kind of stop here. We're just doing one round of this. So we also tried changing different parameters. So I talked about imputation and how that can influence the kind of sort of classification that you try and do, because it can introduce bias to basically emphasizing class distinctions that weren't there. It's just that the way you're imputing these values doesn't consider for that there may be biases in these kind of missing data. So we tried different variations of that, but it seemed like it's fairly robust to that, which is good, which indicates, it's fairly robust to certain parameters that we're using. Various filtration of the univariate filtration, of various parameters on the univariate filtration. So at least in this context, a p-value on FUR of 0.05 is relatively arbitrary, because we're not really trying to call statistical significance. We're just trying to set a threshold on this that, we think anything above this are our most interesting features. But it's also worth looking at effect size at log-fold changes. So when it comes to actually developing a test, we're probably quite interested in at least detecting proteins that do vary a fair amount, relating to this issue of the limit of detection, where, if they're small changes, although they might be biologically relevant, they probably don't make that good practical biomarkers if, say, the variation in their abundance is less than stochastic variation of the instrument you're using or the method you're using to identify them. And then we also changed some of these kind of thresholding runs. So 75 repeats is relatively arbitrary, so we also tried a few there. And then the number of splits is relatively arbitrary, so we tried a few there. But it seemed fairly consistent in these. It seemed fairly robust and very consistent against these kind of parameters. So then, kind of doing some kind of more. Filtering through those and choosing for optimal models after that, we ended up settling on this set of 30 proteins. You can generally see, again, it's always worth this kind of stuff, it's just eyeballing the data, so basically saying, it can give you an idea if your model is just picking up on complete incoherence, but you can see that some proteins are kind of performing quite well here. So if you see this guy in here in orange is doing quite a good job at separating the orange category from the other categories, somewhat similar for that one as well, and a little bit for that one, but you can also find cases where it's not like certain proteins distinguish between maybe some of the groups, but not all of them. So you can see that there's this more kind of complex signature going on here that's not just like you're looking for individual proteins that completely associate. You've got some that are a bit more complicated. So like that one is separating maybe that one, but also the healthy samples down there as well. Same for this one where you've got the CNO and then the infectious osteomyelitis separating there as well. So there's this kind of more complex relationship, which hopefully we picked up on a bit better here by focusing on these proteins. And so this final thing that I did, so the PC on the left is just the PC I've showed initially working on all 487 proteins. And then on the right is just basically doing the PC on all samples, but kind of subsetting to these 30 proteins. And the idea is you can basically see here that by focusing in on these proteins as subset, we've effectively discriminating between the classes a lot better. Not perfect, so basically where there are, where you've got where you've got data points labeled, samples labeled, that represents where there's been a misclassification. The name that's shown is the right classification and the color is the wrong one. So it's not doing perfectly, but generally the CNO samples here in orange are doing quite well at separating. But it's not doing such a good job at separating the other classes, which was one of the goals, but the main goal was trying to separate the CNO samples, and that seems to be performing okay here. And yeah, so the stage we're at now is, so the idea isn't necessary to kind of build this model and then use it to kind of predict outcomes or predict new samples in the way that, we would use this kind of random forest that we trained on this data. Instead, it's to kind of take forward these proteins. So it's a little bit different. We can be a bit more flexible in terms of, not needing one big model that kind of performs quite well. But what we're going to go forward and do is basically investigate these 30 proteins, try and test them via some kind of framework as well, but also use kind of more orthogonal testing as well. So I was talking about kind of more low throughput methods that tend to be more accurate. So these would broadly be things like assays. So this is quantifying proteins via more kind of classical molecular biology methods that are based more on kind of like the biochemistry than actual kind of high throughput approaches. Generally, say more accurate, but more low throughput. So we're going to test these in a set of both the set of validation data we identified previously, so the stuff we held out, but also a set of additional data. So one is an adult data set. So this has all been performed in pediatric patients, but we also have CNO and healthy samples for adult patients, and we're trying to see if these proteins are consistent both for pediatric patients and adult patients. But we also have longitudinal data. So these are patients that start off naive to treatment and have been treated with certain therapies and then to help with this condition. And the idea is to see if we can predict decreases in disease severity with these 30 proteins. Are these directly related to the actual severity of the disease as well as the actual, distinguishing the disease itself. That's where I'm at. Thanks for listening, guys. Thanks to my CBF members. Thanks to Project Collaborate. Thanks to Alex and Liverpool for inviting me to speak. You guys got any questions?
Thanks very much. Yeah, so as usual, we'll take questions in the room first. Anybody online has a question, either drop it in the chat or raise your hand and I'll call you. Yeah. Any questions from the room? If not, I'll give my kind of traditional open gambit. So for the first slide where you reduce from 187 to 30 proteins, what's the risk of overfitting that? Can it be that you have a small sample size, you've just found the 30 proteins that happen to give you the result you wanted.
Yeah, so that is exactly the issue. So that's one reason we're trying to use this kind of more orthogonal set to compare against. But it is a huge issue with this kind of data set. You know, a lot of biomarkers do end up kind of having issues of, you know, you're predicting great within this subset. Maybe you're predicting within a population, but you saw at the start. Sorry, I gotta go through my slides. Yeah, so there is an element of site specificity to some of these samples. So we have tried some, I don't have time to go into various methods to deal with this, but it might be that we're struggling to separate, you know, just so this is the Dresden sites, the physical location where these samples collected. It might be that we're overfitting there as well. So this is the kind, I guess, kind of the crunch point is how well it performs on this kind of validation data.
Yeah. So what was the I guess this kind of goes into what the CBF does more widely as well, but what's the genesis of this project? So you're a CBF in Liverpool. People approach you and say, we are a health institution, we have an interesting problem, would you go and look at it? Do you propose something to them? And in particular, in this case, you know, you're saying you're working with Liverpool, but all the target data is in Dresden. Yeah. So what's the structure there like?
So it can depend. So it is with a prior collaborator we've worked with a bit. So it's Christian Hedrick at the Alder Hey Children's Hospital. I don't quite know how that link started off. You know, it's kind of based on someone. So Eva, the manager, I think had links with him from previously. But the general idea, I guess, you know, kind of business model if I apply that approach, though that terminology is like, someone may have a kind of a research question they want to ask. They think, you know, doing some kind of bioinformatic analysis, something like that we can offer is the best way to do it. Their options are either, you know, find a more kind of individual bioinformatician, someone who, you know, a professional bioinformatician, not as part associated with the group, not associated with the research, and though, you know, will probably be quite relatively expensive compared to us because because they're kind of independent, but also like the sort of general expertise that we have as kind of an individual organization that we can offer. Or the other end scale is kind of hiring an in-house postdoc to then get trained up, but there also comes risk with that as well. So that's the kind of general idea. Generally, I think a lot of it's kind of advertisement, people knowing about us as well, you know, is how we kind of get out there. I think there was a question, so.
Yeah, sure. Do you want me to read it out? Yeah. So Nelson asks, do you find the issue of data collision when performing the analysis in multidimensional data? If so, how do you deal with such issues?
What's data collision in this context? Yeah, sorry. It can be an issue with this kind of field as well, is some of the terminology can vary.
Yeah, so I suppose we'll give Nelson a second to explain data collision. Any other questions in the room?
Say in this slide, and you're collecting samples. Is it you're only allowed to collect samples for that one particular study, and say, for example, after that they won't be destroyed or never used again? Or can you sort of form a database with these? So is it one of those things where, you know, they have to, you know, whoever sample it is, you know, they have to agree to the first study, and then they want to be a database, they agree to the second, and so on. That's it.
Yeah, so it depends. Yeah, it depends. Yeah. People can agree, you know, I'd only want this sample to be donated to this one study, but then chucked away. Other times, no, it's like, we'll take the sample. Yeah, so you have this concept of biobanks. So it's the idea that, yeah, you have this kind of build up of samples and people are able to use for different sets of research. Obviously, you're constrained by the physical amount of sample that you have, but generally that does seem to be something people are moving more towards, because I don't think I was able to show it, but the kind of dates that some of these samples were collected is quite broad as well. So some of these samples have been present basically in the freezer for quite a while now as well, which is a whole other issue of technical variability as well, but I don't have time to go into it. That's it.
So
characteristics.
yeah, it's data collision apparently when you have data that share characteristics amongst
Oh, okay. This is kind of
to
by doing features with data. So is this the issue when you're doing kind of multivariate selection? You know, things that are basically correlated. People have to pick one of them in that kind of group. So this hope. If I'm answering the question, you know, I'm not just kind of leaving it. That's one hope of doing that 100 rounds of lasso regression, is you're kind of picking up on some of that inherent kind of, you know, it's sometimes almost doing a coin flip in which it's whether it's picking one correlated protein or over another. Sorry, I hope that's answering your question. I didn't mean to kind of straw man it.
Okay, any other questions online or in the room? feel like everyone always gives me the opportunity to ask dozens of questions. That's fine. So you were talking about the limit of detection problem. Why wouldn't you just put those in as 0 or put them in as some value below what you think your limit of detection is? If you just say it's unknown, doesn't that potentially give it the opportunity to be viewed as potentially being above the limit of detection?
Yeah.
If you say it's missing, you're not saying you're not making use of the information that you have, which is it's below this threshold, I think.
Yeah, so it depends on the analysis you're doing. So generally you want to try and not impute stuff because you are, basically to the TLDR inventing data, right? You're kind of you're using this issue of kind of applying a parametric model is right. You're kind of assuming I'm going to assume this data is like this because of this model. But whether that model you're using is right or not is a big question. You can there are some methods. So I think I've got min-det, that min-det method, that basically is just inferring, a lower quantile of that entire data distribution there. That can be okay, and as it turned out, that was absolutely fine. But I think the issue you can get is the differences between reasons for the missing values. So you have, and the one that's potentially the biggest issue is when it's not randomly missing. So it's not randomly missing and it's not due to limit detection issue as well. So it's not about the actual abundance of it that's the issue. So an example would be, say if this protein is potentially missing specifically because of the biological context, that itself is some kind of confounding, right? And if you kind of impute without considering it, the flip side of that, and it's something we looked into, is that's also really interesting. If you assume that limited detection issue is the reason it's missing, then that's quite interesting because it means it's potentially quite poorly abundant consistently in those samples. But yeah, it depends. When doing this, at least the sort of methods we did for this multivariate classification, this multivariate feature selection, required no missing data. But there are other methods we could have used to model that specifically.
Okay. Just maybe as like a final big picture question, what do you think is the limiting factor in bioinformatics at the moment? Is it the amount of data? Is it the quality of the data? Is it the compute power? Is it the models that you have?
I think it can depend. I think it can be the amount of data in some ways that it's quite. Data can be very specific. So I talk about this issues of sites. What that broadly introduces is what's known as a batch effect, right? It's technical variability that compounds with your data. It might be that there's plenty of data out there that would be perfect, if not for certain technical factors. So it relates back to what I was talking about with data generation being difficult. Because of that, it's really suffers from inherent technical variability. And it can be as simple as one guy does this analysis on one day, someone else does it on another day. That's going to introduce variability. So the data kind of is there. It's just not consistent, and it's not. It's not consistent, and it has kind of confounding. So whether it's actually of any use to the question, it can sometimes be the issue. And then I think the other side is more kind of capacity to process the data and work with it, because there are, as I say, huge databases of genetic variants, but hundreds of millions, and it's either just individuals working on it, enough people working on it, or the infrastructure to work with it, enough high performance computing suites, that kind of stuff, and expertise as well. So there are issues of bioinformatics, as I say, kind of a lot of people kind of developing methods, occupying kind of in a vacuum. A lot of people kind of reinventing the wheel when there are a lot of other disciplines that basically already done it. I went to quite an interesting talk, was talking about so spatial transcriptomics. That's trying to basically, if you think of a bit of human tissue, you're taking a slice of it, you've got different cells spread out spatially within that tissue. It's trying to investigate how those cells and how the genes expressed and proteins in that cell are varied. People have tried to develop methods to do that. That's come out about 5-10 years ago. That started to be developed. People started to develop methods, but actually a lot of the methods that were developed in geospatial analysis apply directly to it. So I went to a talk with a guy that was trying to kind of merge those two fields. So I think that's quite interesting. So I guess it's both a problem and a potential solution as well.
Yeah, I mean, just looking at those PCA charts, I'm sure there's any number of our students here who are working on very similar things in there.
Yeah. It's not a bioinformatics talk unless I bring up the PCA.
Yeah, it's really interesting because, like in this plot, talking about the batch effect, the only link that you've got is healthy people in Liverpool and in Vanderbilt, and neither of those two have got any connection to Dresden. So, I mean, the only thing you can really do is say, do batch effects exist, and try to compare Vanderbilt and Liverpool's healthy people and say, do they look roughly the same? And even if they do, you don't know how that's going to impact you with Dresden though.
Yeah, so that's one element of plotting the data, was to say I kind of split the healthy samples up. The way I did the univariate filtration was also a bit of an abridged version. Also, I'll stop sharing. There. Yeah, so oops. Sorry, yeah. So I was looking for differences within Dresden and then overlapping them with differences between as well. So trying to not let that site set separations impact it, but then it potentially overfitting to Dresden as well. So we did do various stuff like I talked about date. We fit models, did this filtration where we considered date and saw how the proteins overlap with that. So of these 30 proteins, yeah, of those 30 proteins that I showed at the end, we have chosen different ways of filtering them as well. So different kind of like, they're sort of different rankings of the proteins, basically, about how important we think they are. So again, we're quite lucky that we don't need to sort of build a model that's dependent on, these statistically selected proteins. We can be a bit more kind of ad hoc with it.
Cool. All right, thanks very much.